Hi biologists, welcome to this screencast that's going to cover chapter 9D of your textbook on speciation. So our learning intention is to learn about the ways in which new species are formed, and to be successful you should be able to define a species biologically, explain what allopatric speciation is and sympatric speciation is. Your study design dot point, you need to be able to understand evidence of speciation as a consequence of isolation and genetic divergence, including Galapagos finches as an example of allopatric speciation and Hawea palms on Lord Howe Island as an example of sympatric speciation. Please note, they've named Galapagos finches and the Hawea palms as specific examples. You're going to need to know those examples, and I'll talk about them here, but please make sure that you do understand them. If they've named them, you're going to be examined on them. So before we start, in terms of speciation, we should understand what a species actually is. So for something to be defined as a unique species, it needs to be able to reproduce and ultimately make viable and fertile offspring. So you need to be able to define viable and fertile. So viable just means they're able to survive, they're not lost before they're born or anything like that. And fertile means they are also themselves able to reproduce. In other words, the offspring can grow to reproductive maturity and produce offspring themselves. But you need to know viable and fertile, and you need to absolutely be able to define a biological species as being able to have viable, fertile offspring. So speciation is the process by which new species are formed. So they'll evolve from an existing species and then diverge. So meaning genetically they grow apart from the original species. Should those two species come back together and try to produce fertile offspring, they'll fail to do so. So, in other words, you have your common ancestor, if you like. It diverges off and produces a new species. If the new species tries to reproduce with the original species, it can't, either because the offspring is not viable, or the offspring is not fertile, or both. If that's the case, it's now defined as a new species. So there's information here. When you get two closely enough related species, for example, the Siberian tiger and the Sumatran tiger, they are subspecies of Panthera tigris. So phenotypically they're different, but they can still breed with one another and produce fertile and viable offspring. However, lions and tigers, they're relatively closely related. So you can take a male lion and a female tiger, and that can produce a liger. But a liger, whilst viable, is infertile. It cannot reproduce. On the flip side, if you've got a male tiger with a female lion, you get a tigon. And so here you've actually got a picture of a liger. So you can see, get the tiger stripes, but very much a lion type body. But that individual would be infertile, unable to reproduce, and therefore that, even though these can technically reproduce and you get these hybrid animals, they are separate species. This is Panthera leo, which is the lion, Panthera tigris, which is the tiger. They are completely different species, cannot produce viable and fertile offspring, and therefore are classified as separate species. Okay, an important concept to understand is reproductive isolation. So I'm going to play this video for you that describes the different types of reproductive isolation, then I'll talk through those with you at the end of the video.
Thanks for stopping by. Today we are talking about speciation. Speciation is the formation of one or more new unique species from a current species. In order for one species to split into two or more, a faction within the species must not share its genetic information with the rest of the species. This is called reproductive isolation, and there are two main categories of contributors to this isolation: prezygotic barriers and postzygotic barriers, so named for whether they occur before or after fertilization. Prezygotic barriers result when two groups within a species isolate themselves from one another in some way. So let's look at some examples. Temporal isolation occurs when members of the same species are active at different times of the day, month, or even year. More specifically, they breed at different times. For example, if some members of a group breed during the day and others breed during the night, they are isolating themselves from one another and not sharing genetic information. The same would be true for one group breeding in the spring and the other breeding in the fall, again, both from the same species. Behavioral isolation is another. In this case, members of the same species behave differently, especially with their mating practices. So these members may have slightly different mating rituals that don't attract members of the other group, thus isolating who they mate with. Others include mechanical isolation, where some members physically can't mate with others, habitat isolation, where the habitat occupied by members of a species differs and thus breeding interactions differ, and gametic isolation, which occurs in aquatic animals. You can learn more about all of these by following the links in the description. Now to post-zygotic barriers. Again, these barriers occur after fertilization has occurred, so mating happens but no viable or fertile offspring is produced. These include reduced hybrid viability, in which case there is successful fertilization, but development is not successful or results in very weak offspring, which is prone to early death. Another example is reduced hybrid fertility, in which case a mating is successful at producing offspring, a hybrid of the parents, but the offspring is not fertile and thus produces no offspring of its own. The classic case is a mule, the offspring of a male donkey and a female horse. The mule lives a full life and can function just fine, except when it comes to reproducing. This is one reason that donkeys and horses are considered to be different species. A final example of post-zygotic barrier is hybrid breakdown, in which offspring is produced, and that offspring may be able to reproduce, but the second generation will be weak and or sterile.
So from your textbook, there's two major categories of reproductive isolation: pre-reproductive or before reproducing, and then post-reproductive. So if you're geographically isolated, you can't reproduce. So it's a pre-reproductive isolation mechanism. You may live in different habitats. Again, you're not going to come in contact with each other. You can't reproduce. Temporal means different times of the day. Things like nocturnal versus diurnal, awake during the day. You're not awake at the same time, you can't reproduce. That one becomes important when we talk about the Hawiya palms. Behavioral, so different mating behaviors, mating calls, or you're just repulsed by their behaviors. And I'll talk about that in a moment. And structural, so you may actually not be compatible with one another. So even if you tried to mate, you just physically can't do it because the bits and pieces don't work. All right, so that's pre-reproductive. It's before you actually reproduce. Let's assume that you've overcome all of those barriers and you've actually managed to attempt reproduction. Gamete mortality, so sperm may not be able to penetrate the egg, so fertilization can't occur. Fertilization might occur, but then the zygote dies, so it won't survive. Or as in the case of the tiger and the lion mating, the hybrid is actually sterile. Same with a horse and a donkey, which produces a mule. Viable offspring, but they're infertile. So that's after reproduction has happened, but it's reproductive isolation. It can't be continued. Those genetics do not continue on down the line, and therefore it's post-reproductive isolation. So pre-mating isolation, we've already talked about. Isolation, day versus night. Isolation in location, so ecological. Anatomy, that's the structural and behavior. So your day versus night, diurnal versus nocturnal. Or you might get something like this guy, who, let's be honest, behaviorally is unlikely to ever successfully reproduce. All right, allopatric versus sympatric. Allo means different, and patric means fatherland or homeland. So like patriarchy is, the patriarchy is the world dominated by men. It's very sexist and is not okay, but that is what the patriarchy is. It sort of tells other people how to live their lives, kind of thing, which I'm telling you that as a way to remember allopatric, different, allo, fatherland, patric. So allopatric speciation occurs where there's a barrier between two populations, different homelands, right? Sympatric, the sim means same, patric is still fatherland. So this time the species diverge from a common ancestor, but there's no geographical barrier between the two. We're going to talk about both. Now, this is a section of video that you've seen before. It's the David Attenborough documentary. I want you to watch it. I want you to think about it as you're watching it. And based on what I've just described, I want you to be able to answer the question, which form of speciation is occurring here? Is it allopatric or is it sympatric?
Wolf volcano. Its giant crater lies exactly on the equator and is encircled internally by steep cliffs. And on its flanks, evidence was discovered of a catastrophe that might, paradoxically, eventually lead to the creation of a new species. This flow of recently solidified lava has created an impassable barrier 100 meters wide right through the vegetation. The fresh lava is razor sharp and almost impossible to cross. On it lie bones. Bleached white by the baking sun, they're those of giant tortoises. Some animals that got stuck here are still clinging to life. Tortoises are tough. They can survive for months without any food or water. And that worsened their fate. It will take them months to die. Why so many tortoises tried to cross the lava barrier, nobody knows. What drove them to keep attempting this impossible journey? Perhaps they were desperately trying to rejoin other members of their group. Their deaths, however, are significant. They illustrate a principle that applies to the whole of nature. It's not unique to the Galapagos, but it's because of the Galapagos that we first came to understand it. This tragic scene, filmed for the first time, may provide a clue as to how a new species may start to evolve. The lava flow created an impassable physical barrier across the tortoise's territory, dividing it into two. So, two tortoise populations that were once one must now live apart. If there is any significant difference now or in the future between their two territories, the tortoises may eventually become two different species.
Hopefully you answered allopatric speciation. And so this is an image that helps show you that. You've got one single population. There'll be some genetic variability in this population, but a single species. A geographical barrier comes up. In the case of the video, it's a lava flow that cooled and then prevented the tortoises from crossing from one part of the island to another. A geographical barrier, isolates the two populations. And then on each side of the lava, you have different selection pressures. Here it's sunny, here it's cloudy and raining. So different selection pressures, meaning different phenotypes are likely to survive to reproductive age, pass on their genetics, and gradually, and we're talking over geological time, it takes thousands or millions of years in order for this to occur. They will be so genetically different that if they came back together again, they can no longer interbreed and have viable, fertile offspring. In other words, a new species has been formed. This is the same concept, but illustrated in your textbook on page 517. Different populations with variation in between. They've used a mountain range as well because all of the textbooks do. There's selection pressures that act on the populations on different sides. So on this side, the red allele was selected for. On this side, the blue allele was selected for. And then given enough time, they become genetically different enough that if they tried to interbreed, they can't. Same principle, just a different illustration of the same thing. This tip is basically telling you how to answer a question about this. It's commonly, very commonly, a question on Saxon exams asking you to explain the process of allopatric speciation. The first step, as always, there's genetic variation in the original population, and then, which they've not included in there, so I've added it, there's a geographical barrier. The environment was different on each side of the barrier, leading to different selection pressures. And then given enough time, there's enough genetic differences that led to them becoming separate species, because if they tried to interbreed, they can't. So in your textbook, it actually tells you how to answer this question, but this first point, always important for getting the first mark in a multi-mark question related to this. So for allopatric speciation, you need to know about the Galapagos finches. Keeping it really simple, you don't need to know a lot. You just need to know them as an example. You need to understand. So the geographical barrier is the fact that the Galapagos are islands. They're separated by water, so the birds aren't just going to pass from one island to another. The selection pressures, different islands had different seeds or different food sources. So therefore, there was a need for the finches to evolve different shaped beaks to crack open tough seeds. Or if you remember the video that we watched with David Attenborough, there's the vampire finch, which actually could penetrate other organisms and drink their blood. Different food sources required a different tool. The different tool is a different shaped beak. And so given enough time with those selection pressures, they evolve different shaped beaks until now, if you were to bring those finches together, they can no longer reproduce. There was originally one species, we think, of finch that went to the Galapagos, managed to cross from South America to the Galapagos Islands millions of years ago. There are now 18 different species of finch on the Galapagos Islands. And this process of allopatric speciation, separation, different selection pressures, and ultimately genetic difference builds to the point where they're now separate species, we think that's the process by which it occurred. Sympatric speciation occurs within the same population and there is no geographical barrier. So there's a couple of different ways that this is proposed. There's genetic variation which has different phenotypes, and then the selection pressures within the same population that act over time, allowing individuals with different phenotypes to diverge from one another to ultimately reproductively isolate and therefore become a separate species. And I'll give you an example of that with the Hawiah palms in a moment. Alternatively, it can be formed by polyploidy. So you should hopefully remember that polyploidy means that they develop an entire additional set of chromosomes. So for us, that would mean instead of having 2 sets of chromosomes and being diploid, you might have 3, or even in the case that I've written here, you go to 4N, 4 sets of chromosomes. And so this is an example from your textbook of that. You've got a diploid organism that instead of meiosis producing haploid sperm and eggs, or haploid gametes, instead of doing that, there's an error in meiosis and the gametes are actually diploid, just like the parent organism. Obviously they fertilize, meaning that you go from having 2 copies here and 2 copies here to having 4 complete copies of chromosomes. So it's a tetraploid organism, or 4N. If that happens, it's obviously going to be phenotypically different. It's also going to be incompatible with the original species, all right, because the original species has diploid. This has tetraploid. They're not going to be able to interbreed. So that's one way you could get sympatric speciation. But let's look at the way the Hawiah palms did it. So there's a video here where a guy talks a little bit about some of the key selection pressures that were at play, and he's doing some research. He talks about that as well. So I'll play that. It does explain it at the start of the video. The rest is more just sort of interest about the work that he's doing.
Hi, I'm Matthew Coto. I'm a PhD student in Speciation Genomics at Imperial College London, and I want to talk to you about the plant phenotyping experiments that I've been carrying out here at the National Plant Phenotyping Infrastructure at the University of Helsinki, all of which has been made possible by an EPPN 2020 Transnational Access Grant.
My PhD research is aiming to identify genes which have facilitated speciation in the Hawiah palms. These are 2 sister species of palm tree, which are found only on the tiny Lord Howe Island off Australia, where they have diverged from a common ancestor due to preferences in soil type and differentiated flowering times. They are particularly interesting because they have undergone sympatric speciation. That is, they have diverged in the presence of gene flow and without any geographical barriers. Strong evidence for this type of speciation is rare, and very little evidence exists of a specific genetic mechanism which could facilitate this speciation. The phenotyping experiments I am carrying out at the University of Helsinki hope to change this. We have identified a series of candidate genes which may have been involved in the speciation process by simultaneously affecting both soil stress tolerance and flowering time in Hawiah palms, but the function of these genes needs to be investigated further. To do this, I'm growing around 2,000 of the model plant species Arabidopsis thaliana at the Finnish National Plant Phenotyping Infrastructure. I'm using a range of mutants with homologs of our candidate Howier genes knocked out, and growing them in the presence of multiple soil stress treatments, such as drought, high salt and high cadmium levels, conditions which mirror those found on Lord Howe Island. The facilities here allow me to impose highly controlled environmental conditions, and allow me to gather an incredible amount of highly detailed phenotyping data. Through automated imaging using RGB, infrared and chlorophyll fluorescence cameras, I've been able to carry out analysis of traits such as plant shape, size and colour, alongside measures of plant temperature and photosynthetic function, which allow me to investigate the fine-scale stress response of a huge number of plants. By combining this soil stress tolerance data with analysis of flowering time, I've been able to identify genes which could have facilitated sympatric speciation in Howier palms, providing key evidence of a plausible genetic mechanism for sympatric speciation. This work would not have been possible without access to these superb phenotyping facilities, made available to me through an EU EPPN 2020 Transnational Access Grant, which I was fortunate enough to hear about through colleagues in my field. Thank you for watching, and thank you to my collaborators at the University of Helsinki for all of their hard work throughout the entire project.
So he touched on it in the video, but let's walk you through it. Again, it's the example named in the study design, so you need to know. Howier palms, we believe, went through sympatric speciation because of a difference in pH in the soil. This species, I don't even know how to say that, Balmoriana, there we go, Howier Balmoriana, prefers neutral or slightly acidic soil, so it has a lower pH. Howier Fosteriana preferred an alkaline soil. But these two species originally would have been one with natural phenotypic variation. So some were slightly more suited by the higher pH, some by the lower pH. So the natural variation occurred in this common ancestor, and then Howier Fosteriana diverged after it managed to colonize alkaline soil. Now this would have happened again over geological time, which is a long period of time. Additional physiological changes developed. One of the key ones is that Fosteriana would flower at different times to the other species of Howier. Right, so if they're flowering at different times, remembering flowers are the reproductive organs of plants, that's reproductive isolation. So one species could no longer reproduce with the other species because they flowered at different times. It's temporal reproductive isolation, different times, and that over time allowed speciation to occur. Those physiological changes and genetic changes built up over time until they were now completely different species. So even if they flowered at the same time, they're genetically different enough now they would no longer be able to interbreed and produce viable, fertile offspring. So hopefully that all makes sense. What you need to be able to do is define a species biologically as being able to produce viable, fertile offspring. Explain allopatric speciation, which is created by a geographical barrier with differing selection pressures on each side of the barrier, ultimately building genetic change and a new species. And sympatric speciation, using the example of Howier palms, where the initial change in pH in the soil allowed for different phenotypes to flourish. Physiological change developed over time, leading to reproductive isolation through differing flowering times, and therefore ultimately speciation of two completely different species. So you need to be able to explain all three things. What is a species, allopatric speciation, and sympatric speciation? As always, if any of that's unclear, or I can help clarify anything, please let me know. Otherwise, thanks once again for watching.